For as long as we've studied life, we've tried to draw a map for it. A big, grand family tree that connects every single creature on Earth. But what if we've had the basic shape wrong this whole time? Yeah, let's get into it.
I mean, it's one of the biggest questions out there, right? How is everything related? From a tiny little bacterium all the way up to a blue whale. And for a long, long time, we thought we had a pretty good answer. For decades, we pictured it as a tree. I mean, you've seen the diagrams, right? There's that single trunk at the bottom, that's the common ancestor, and then all these branches just split off and off and off for every species. It's a really clean, elegant idea. It just makes sense. But new evidence is suggesting the real shape is something else entirely. I mean, what if that nice, neat tree is just too simple? Back in 2009, a paper came out that basically dropped a bombshell on this whole idea. It suggested the map of life isn't a tree at all. It's something way weirder, and honestly, way more interesting. All right, so before we get to the new stuff, let's just quickly go over the model that's been the standard for decades. This is the official map we've all been working from. Now, our understanding has changed over time, for sure. You might remember learning about the 5 kingdoms in school, plants, animals, fungi, and all that. But we've since moved on to something a bit more fundamental, something based on deep genetic data, the 3-domain system. So this was developed by a guy named Carl Woese back in 1990, and he did something brilliant. He looked really deep into the genetic code, specifically at something called ribosomal RNA. Now, you can think of RNA as like a crucial piece of machinery in the cell's factory. It's in all living things. By comparing how that one piece of machinery was built across different species, he found these massive fundamental splits in the tree of life that nobody had seen before. So the key here is, this whole system is built on genetics. And you can see the main differences right here. You've got bacteria and archaea. Now, on the surface, they look kind of similar. No nucleus, single circular chromosome. But on the inside, chemically and genetically, they are worlds apart. And then you have Eukarya. That's us, and plants, and fungi, and pretty much everything you can see without a microscope. We're a whole different beast. We've got a nucleus, multiple chromosomes, the whole shebang. So, this 3-domain system became the foundation for our modern tree of life. For over 30 years, it's been our guide, our roadmap for understanding evolution. And it's been an incredibly powerful tool. It has served science so well. But maybe it's time for an update. Okay, so now we get to the really interesting part, a pattern in the proteins. A totally different group of scientists decided to come at this from a completely new angle. Instead of looking at one single gene, they decided to look at proteins. And this really gets to the heart of what science is all about, right? Asking that one big question. But what if we've been reading the wrong map? What if the very foundation we've built everything on is just a little bit off? So, here's what they did. Remember how the old model looked at one gene, that ribosomal RNA? Well, this 2009 paper proposed looking at a totally different data set, the length distribution of all the proteins an organism makes. Not just one, all of them, thousands and thousands of them. It's kind of like taking a statistical fingerprint of a species. The pattern of all those different protein lengths creates a unique signature that tells a story about its entire evolutionary past. And their conclusion wasn't some minor tweak. It was huge, a total reimagining of the entire structure. They came right out and said it. The global taxonomy of life can be illustrated as phylogenetic circles rather than phylogenetic trees. Circles, not a tree. Okay, that might sound a little abstract, so here's a great analogy they used. Think about how astronomers study stars. They don't just follow one star through its entire life. That would take billions of years. Instead, they map out thousands of stars on a chart based on their brightness and temperature. And by doing that, they can see the entire pattern of a star's life, from birth to death, all at once. Well, this research is basically doing the same thing for life, but instead of using starlight, it's using the data from thousands of proteins.
So, life in a circle, what does this new map even look like? Well, get ready, because this is probably not what you're expecting.
Okay, so first, here's our old friend, the classic phylogenetic tree. You've got the 3 big branches, bacteria, archaea, and eukarya, all sprouting from a common root and just branching outwards and upwards. It's a story of simple, linear progress. We thought life looked like this. But the amazing thing is, when you take that protein data and plot it out, that tree shape, it just dissolves, it vanishes. What you get instead looks more like this. Boom, there it is. 3 separate, distinct circles. One for Eubacteria, one for Archaea, and one for Eukarya. In this model, species aren't on these branches that are just stretching out forever into the future. Instead, every species is a point along the edge of its domain's circle. It's a closed system. It's constrained, not endlessly expanding. So these are the phylogenetic circles. And the theory here is that life within one of these big domains evolves as a kind of constrained community. Species exist on the edge of the circle, not on these endless branching paths. And that implies something massive. It means evolution isn't just a free-for-all with every species for itself. It might be more of a communal thing, with some kind of global rules that limit or constrain how species can develop. They're all evolving together inside a shared boundary. All right, so a new model is cool and all, but it's only really useful if it can explain something that the old one couldn't. And as it turns out, this circle idea might just solve a really old evolutionary mystery.
So what? Why is a circle a more powerful idea than a tree? I mean, apart from just looking cool, what does this actually tell us about how evolution works?
Okay, let me introduce you to something called the C-value enigma. For decades, biologists have been scratching their heads over this. It turns out that the size of an organism's genome has pretty much nothing to do with how complex it is. Which is nuts, right? Just look at this. An E. coli bacterium has about 4.6 million base pairs. A human has 3.2 billion. Makes sense so far? But then you have an amoeba with 670 billion base pairs. It's absolutely massive. And the classic tree model has never really had a good way to explain why this is.
But this circle model, it offers a new way of thinking about it. It suggests there's a kind of bidirectional force in evolution, a push and a pull. Sure, there are pressures for a genome to get bigger and more complex, but maybe there's also a constraining force from this evolutionary community that pulls it back, keeping everything within the bounds of that circle.
So what are the big takeaways here? 1, evolution might be more of a communal thing inside a domain than we thought. 2, there might be these global constraints on how species can evolve, rules we never even knew existed. And 3, this new map, this new way of looking at things, could help us solve a lot of other long-standing puzzles in biology.
Think about it. If the tree of life, one of the most famous icons in all of science, is actually a series of circles, it really makes you wonder, doesn't it? What other fundamental truths of biology are just sitting there, waiting to be reshaped? What else have we been looking at the wrong way, just waiting for someone to come along and see the pattern hidden in plain sight?